My name is Joseph Wiemels, and I'm a professor at the Center for Genetic Epidemiology at the University of Southern California. My research examines the causes of childhood cancers, including leukemias, brain cancers, and sarcomas. Today we will consider how chemicals may cause childhood cancers. Every cancer cell exhibits a suite of characteristics that serve to distinguish it from normal cells. These features ensure the growth and immortality of cancer, and typically 5 or more mutations or changes are needed for the complete conversion of normal cells to cancer cells. Interestingly, though, during embryonic development, body tissues in their normal state possess many of these same hallmark features. For instance, blood-forming cells are essentially immortal and continue to produce new cells throughout life and can invade other body tissues at will. In addition, all body tissues exhibit embryonic status and rapid cell division in early development. So, for these reasons, childhood cancers tend to exhibit fewer mutations than adult cancers. Now, let's talk about the way that mutations are formed. For more than 50 years, scientists have recognized that chemicals that react and change our DNA can act as carcinogens. Hydrocarbons created from combustion are classic carcinogens, along with aromatic amines and nitrosamines from tobacco. These chemicals and others bind directly to DNA, leading to a change in the code, which can contribute to the cancer phenotype. Here, on the lower panel, we see that nitrosamines and their decomposition products can directly bind DNA, leading to DNA mutations and directly contributing to cancer. After the universal acceptance that mutagens are carcinogens, scientists have discovered many chemicals and pathways to cancer that are not directly related to DNA damage. Chemical and non-chemical stressors may disrupt hormone signaling pathways, cell communication networks, immune system, and inflammation. This wide array of activities of carcinogens may help explain why cancer rates continue to rise despite our increased attention to such effects, and the reduction of mutagenic chemicals in consumer products and reduce tobacco usage. Our ability in society to produce and use new chemicals far outpaces our full understanding of their toxicity and effects, and our capacity to regulate their usage. This has led to a more comprehensive perspective called the key characteristics of chemical carcinogenesis. Here we show on the left those characteristics that involve change of DNA sequence or regulation, and here at the bottom of the slide, oxidative stress and inflammation, and along the right-hand side, we consider activities that disrupt normal cell growth or senescence signaling pathways. Carcinogens that impact childhood cancer may include any single one or all of these pathways, but let's consider one in more detail and explain why children have a higher sensitivity than adults. This is the inducing epigenetic aberrations characteristic, which includes changes in the addition of methyl groups on DNA around the genome. Now, DNA methylation is a key aspect of early development as well. Primordial germ cells, or developing ova and sperm, pass through a wave of demethylation and then remethylation of DNA prior to fertilization. Then DNA gets demethylated again in the first days of the zygote, apart from those loci that are parentally imprinted. But by the end of the first week, DNA again is remethylated in all normal cells and maintains this status throughout life. These critical waves of DNA methylation prior to conception and during the first week of life represent a point of high vulnerability to chemicals and nutrients that are necessary for healthy development. All of these changes typically happen before a mother even knows she is pregnant. Now let's take a deep dive into one well-known carcinogen, tobacco. Tobacco needs no introduction as a carcinogen for adult cancers, having a strong role in lung, bladder, pancreas, and other cancers, but less well characterized is its role in childhood cancer. We'll take a look at what childhood cancers are related to maternal pregnancy exposures of tobacco and an interesting mutational consequence of tobacco exposure in childhood leukemias. First of all, what does the epidemiology say? The many investigations of tobacco and cancer have yielded mixed results, and many studies suffer from control selection and recall bias because pregnancy exposures were assessed after the children got cancer years after the pregnancy exposure, as assessed in typical case control studies. On this slide, we see results from a different kind of case control study This one taken within the California birth cohort, where information on tobacco exposure was obtained prior to disease from the birth certificate, and controls were matched to cases without active recruitment. The number of cancer cases was small and showed no tobacco relationship to leukemia, but the study did show cancer risk for gliomas and quite strongly for retinoblastomas. A meta-analysis of leukemia case control studies seems to back up the result from the California cohort study. Here, the summary risk ratio from several studies was 0.98, indicating no apparent risk of leukemia via maternal active pregnancy smoking. However, on the right, we do see that there was an increased risk of children from Latina mothers when pulled apart from other ethnic groups. The plot thickens when we look at paternal tobacco smoking, for which there appears to be a clear dose response. That is, a greater number of cigarettes per day, either before pregnancy or after birth, is related to risk of leukemia in the offspring. Because smoking is a highly addictive habit, the same fathers will likely be exposing their family throughout this entire time period. Now we ask, should household smoking be the culprit? What aspect of tobacco exposure is causing this risk to children? Or is it simply mutational burden the way adults get lung cancer from tobacco? An interesting clue was offered by a study published decades ago, examining the mutational pattern introduced to T cells by tobacco within a marker gene called HPRT. This gene is located on the X chromosome, so everyone can only express one functional copy. If one mutation occurs, the cell becomes resistant to a drug called 6-thioguanine, and we can grow the T cell clone and sequence the mutation. In the presence of maternal exposure to tobacco, the mutational pattern shifts to one that is related to VDJ recombination. These mutations include deletions and the presence of RAG recombination site sequences at the breakpoints. We see that most of the mutations in smoking mothers have this pattern. What is VDJ recombination? Our body cells involved in acquired immunity include both B cells and T cells. These cells can mutate their own genomes to produce a very diverse repertoire of antibodies and T cell receptors. The three classes of genes listed here provide our immune system the capacity to react with any new infection that comes along, including ones which humans have never encountered, such as COVID-19 prior to the year 2020. It is the RAG genes that produce the combination of VDJ fragments that go into making novel receptor and antibody genes, as shown in the lower part of this figure. And they do this using recombination site sequences, which are arrays of nucleotide sequences found throughout the genome. While RAG genes are meant only to target those sequences in specific immune receptor regions of the genome, sometimes this activity can go off target to a gene like HPRT, and tobacco stimulates this illegitimate activity. This provided us a novel hypothesis in the way that tobacco may be causally related to childhood leukemia via secondhand smoking during childhood. We know that gene deletions are a major mutation type in childhood leukemia. Most leukemias have 2 or more deletion mutations in the tumor cell. Could these be related to off-target RAG-induced deletions enhanced by tobacco exposure? Indeed, this seems to be the case. When we measured tobacco exposure using a polyepigenetic smoking score, which is a composite score of several DNA methylation marks sensitive to tobacco, we found that numbers of gene deletions are significantly associated with greater tobacco exposure, shown in the top part of this figure. In the lower part of this figure, we use self-reported questionnaire data on tobacco use to show that the strongest linkage of tobacco to gene deletions was during the time of breastfeeding, which is after birth, and when we know that these gene deletions occur. Additional data not shown here is the presence of cryptic RAG sequences which we have found near deletions in tobacco-associated leukemia tumors. Finally, some takeaways from this talk. First, we note that cancer has many hallmarks, and children have a head start in this process by harboring developing tissues. Second, we now recognize that carcinogens come with many different activities, some of which are related to growth pathways under active changes in the fetus and embryo, making these exquisitely sensitive time periods in development to cancer. Third, while we recognize tobacco as a cause of many adult cancers, it has now been linked to childhood cancers as well and includes specific mechanisms of mutation confined to developing blood cells. Because of our ability to measure tobacco exposure and its effects, and its ubiquity in our environment, we are able to connect this specific mutational mechanism to tobacco. But we note that the inexorable rise in childhood cancer rates over the past decades implicates the involvement of many other environmental chemicals in cancer causation, even while we see tobacco usage decreasing in our society. It is our mission and role to discover how environmental characteristics can impact cancer causation so that we can change and reduce their impacts on childhood cancer for the future.